We have two presenters today. Sara Carillo is the Application Development Team Leader at NIBRT, the National Institute for Bioprocessing Research and Development. And then Ashley Bell is the Field Applications Manager at 908 Devices. So Ashley is going to start, and she will then hand over to Sara, and I'll come back at the end for the questions. Ashley, over to you.
Thank you, Paul, and thank you all who are here. It's my pleasure. As Paul mentioned, I'm with 908 Devices, and I'm here just to give you a very brief overview of what ZipChip is and what it does. So where I'd like to start is just with biology, which we know is really complicated. And when it comes to the characterization, discovery, and development of biopharmaceutical proteins, it's especially complicated, which we all know. So in order to build some of these products and characterize them, we have sequence variations and structure variations to consider. We have post-translational modifications that can happen for a variety of reasons that we have to both characterize and monitor as potential critical quality attributes. And then we have to stress it in order to make sure that these things aren't changing in a way that is detrimental or causing an effect to the immunogenicity, and that our molecules are safe. So in order to do that, we have a wide number of tools available to us that we use, all of us, on a daily basis in this industry. And depending on the attribute, that can be anything from ion exchange chromatography, or CZE for charge heterogeneity. We do a lot of reverse phase HPLC and HPLC mass spectrometry for anything from identity all the way to glycosylation profiling, carbohydrate analysis, purity, sizing, and last but not least, activity. So where ZipChip really fits into this picture is as a simple alternative to many of these attribute characterizations. So we can use a single platform to get at charge heterogeneity, identity, structure, carbohydrate, purity, or sizing, and if we wanted to, in a single day. So instead of being complicated, it's pretty simple. It's about the size of a shoebox, and all of the separation that takes place inside of this box. It fits directly on the front end of your thermal mass spectrometer in exactly the same way as your existing ESI source. You take the ESI source off, you put the ZipChip on, there's no further alignment, and you're off to the races within about 30 minutes or less. The heart of the technology is inside of the box itself in the ZipChip. All of the separation takes place on this chip itself. So what is it? It's a liquid-only microfluidic separation on a glass chip about the size of a microscope slide. We take this glass chip and we etch a microfluidic channel onto the chip itself, through which our analytes migrate in a positive electrophoretic direction directly into the MS inlet through an ESI emitter that is integrated directly into the corner of the chip. We coat this channel with a surface coating in order to ensure efficient separation, as well as prevent absorption and suppress electroosmotic flow. There's no further optimization for the ESI. This is a true nano ESI operating at about 150 nanoliters per minute directly into the source. So at the end of the day, the entire workflow is pretty simple. You would choose an application and decide what the goal of that application is, which would then dictate a kit, and you would choose a chip, whether you would like high resolution separation. You might choose a high-speed chip if you're more concerned with throughput, or for native charge heterogeneity and other native protein characterization, you might decide on a native chip and would prepare, separate, and analyze your samples in as little as 2 minutes. So that's a very brief overview of our technology, and to really understand the power in this small package, I am going to turn it over to Sara, who has real data, which we're all waiting for.
Perfect. Thank you, Ashley, and thank you, Paul, for the introduction. So biopharmaceuticals production is in continuous growth with an increasing number of products entering the market, like biosimilars, for example. As well, biomanufacturing techniques are continuously evolving, also bringing big changes such as the introduction of single-use devices, for example. This is forcing bioanalytical technologies to a constant comparability exercise to prove no changes are introduced in the final product, and to obtain always more structural details that allow to confirm drug's identity. This is, of course, required that methods can have an increased dynamic range to spot biotherapeutics microheterogeneity, but at the same time, the methods should be robust, reliable, fast, and possibly automated. The ZipChip front-end separation device has been used in our group in NIBRT for a number of workflows, and they range from intact mass analysis, peptide mapping, and also spent media analysis for cell culture monitoring. As Ashley said before, the configuration and connection of the CE device is straightforward, and it clicks on our Thermo Exactive Plus mass spec as a standard ESI source, and it uses Excalibur software for data acquisition. While the settings for the sample load, injection, and voltages are controlled by the ZipChip interface. On the other hand, the choice of the Thermo Scientific Exactive Plus comes from the need of a flexible platform that could really perform both intact native and small molecule analysis at the same time with the same excellent performances. In particular, the biopharma option available on the Exactive Plus allows to switch on the high mass range mode that enables the acquisition of MS data up to 8,000 m/z, which is required for the native analysis of monoclonal antibody. Of course, each workflow has different kits that are available through the 908 device, and they can be used for the native analysis, for peptides, and for metabolites. And they also include the specific BG for each workflow. As mentioned before, biologicals are very complex molecules, and there are a number of features that need to be monitored to evaluate and comply with market regulations, and also to preserve the drug function, stability, safety, and efficacy. Some of these variants usually are monitored in monoclonal antibodies and are presented here in the slides. For example, we have primary sequence amino acid point mutation. Another example is N-terminal truncation or elongation, C-terminal lysine clipping or the glycine clipping, so the proline amidation, formation of aggregates or the correct assembly of disulfide bonds, as well as some amino acid side chain modifications, like the amidation, oxidation, glycation, or the N-glycans present on the Fc region. Some of these features can really alter the superficial charge of the monoclonal antibody, causing a shift to more acidic or more basic pH of the protein PI. Few examples are presented in this slide. For example, C-terminal lysine is usually truncated in therapeutic mAbs, but when this is present, will provide a basic shift to the PI. Another example is given by Fc and glycan sialylation that brings to the PI to more acidic values. Another important modification that generates charge variance is the amidation. And in this case, both the final deamidated form and the succinamide intermediate will affect protein PI. It is clear that a complex profile may arise from the co-presence of all these modifications. So charge variant profile is considered product quality attribute and is monitored to assess product quality and safety. Charge variant profile is routinely performed on C or LC platform, but it usually returns only a profile with no structural information on the separated species. The ZipChip device is probably the best example of how CE could be easily hyphenated to mass spectrometer with a quick analysis and low sample consumption. For the sample preparation, we only perform the buffer exchange of the protein with the native antibody diluent that is provided in the ZipChip native kit. And this buffer exchange step is also used to reach the right protein concentration that should be between 0.5 and 1 milligram per ml. As I said before, some settings need to be adjusted for such as sample load and the CE separation. And you can see them in the slide here in the screenshot of the software, where we were using basically a default method that is already comes with the software for native antibody analysis. We choose to perform the analysis for 15 minutes to give time to the sample to be completely pushed through the microchannels, and found that these settings worked well with all the monoclonal antibodies that we tested so far in our lab. It is worth mentioning that the sample concentration used in this experiment translates into only 1 nanogram of protein injected, and this makes this platform ideal for samples with a limited availability. For native analysis, the following settings were used on the Exactive Plus, and as you can see, the HMR mode now is on, and this extended the mass range to 8,000 m/z. And for a good compromise between the mass resolution and sensitivity, we used the 35K as resolution settings, and these parameters, again, were used for several monoclonal antibodies without any further optimization. As an example here, I show the nice separation that we obtain in our lab for the electrophoregram of cetuximab drug product and the charge envelope relative to the most abundant charge variant that is located, as expected, between 4,500 and 7,500 m/z. Next slide shows a comparison between the data obtained on the ZipChip platform and the cation exchange chromatography, and these were using the same instrument and settings for mass spec data acquisition. As expected, the elution order in CE is opposite to what is obtained in CX, and we obtain in both cases 9 proteoforms that basically were differing for the content of C-terminal lysine, sialic acid, and so on. This experiment really proves that cation exchange chromatography and ZipChip MS platform can provide orthogonal selectivity that really enhance the confidence in the annotation of the resulting native mass spectra. Another example in the next slide is given on trastuzumab drug product, and the data generated here were convoluted using Biopharma Finder software. And we were able to identify 52 protoforms arising from C-terminal variants, the amidation, succinamide formation, as well as sialic acid content. Thanks to the excellent performance of the QE+, it was possible to identify each of these proteoforms with high confidence, obtaining delta mass values below 30 ppm, even on proteoforms with abundances as low as 0.01%. While the charge variant separation enables the identification of near isobaric species that have now different migration times, such as those containing the amidation or isospartic acid. Moreover, it was possible to see, if you look at the zoom in the bottom right corner, that each peak presents an intrinsic heterogeneity related to the N-glycosylation of the FC region. So is related to those glycans that are not charged and so are not able to shift the protein PI like the sialic acid does. So considering this huge sample complexity, I think it's really a key advantage to have the C or the LC charge variant separation before the MS analysis, respect to what it's usually done with the direct infusion or with the reverse phase intact analysis. The second workflow that we implemented on the CE-MS platform is peptide mapping analysis. And this analysis is considered a gold standard in biopharmaceutical and proteins characterization, as it allows to monitor several attributes in the same analysis, and is considered orthogonal to many techniques. In quality control laboratories, the analysis relies on the fingerprint match of the UV traces generated by tryptic digest and when they are separated on a reverse phase column. However, this analysis usually requires long sample preparation and overnight enzymatic digestion step, plus long analysis time. For sample preparation, we used the automatic sample digest on with the Thermo Scientific Smart Digest kit using the magnetic resin option. Then we cleaned up the sample on a CAT-18 cartridge and loaded the vials on the autosampler for direct injection the CE device. We used the default method provided for peptide mapping on the ZipChip software, so everything was done automatically for sample injection and CE separation. And we decided also to switch on the field strength mode from constant to gradient. I think the gradient option, it's a really interesting mode, as complex samples like tryptic digest will have a range of different peptides, with some eluting too quickly with high field strength, but there will also be some other peptides that are eluting later. And so this can really give broader peaks that can affect the analysis. With the field gradient, it's possible to reach something like a compromise with the early eluting peaks well separated and the late eluting peaks having a nice peak shape. This is an example in the slide performed on the BSA peptides standard mix. But we obtained the same results on bevacizumab drug product. Here I show the full MS electropherogram obtained after trypsin digestion. And during the analysis, also MS data were acquired, allowing confident identification of the tryptic peptides. The separation is obtained in 10 minutes, and the total runtime was around 16 minutes, also considering the sample delivery from the autosampler to the CE device. So this also makes this workflow really an incredible instrument for high throughput. To check data and sample quality, we searched the CE-MS data on the Biopharma Finder software, and it was easily possible to get 100% sequence coverage. Spent media analysis is another important analysis and is an important step also in the upstream process optimization, as the media component may strongly influence not only the cellular growth and the health of the cell, but they can also affect the product titer and the quality. As an example, there are cells that are going through starvation of some amino acids, and this could result in the sequence variant in the produced biotherapeutic. And this, of course, can also alter the product quality and stability. For this reason, it is important to continuously monitor amino acids concentration, possibly with an on-time, an at-line, or an in-line measurement. Usually, spent media analysis is not straightforward, and this is due to the complexity of the sample matrix, and this can require lengthy sample preparation and lengthy analytical methods. With the CE-MS platform, the amino acid analysis was performed directly from cell culture after dilution of the media with water first, and then with the diluent provided in the metabolite kit. Then the default method for the metabolite was selected using only 3 minutes of analysis time, as you can see from the bottom part of the slide. The analysis was first performed on a standard mix to optimize CE and MS conditions, and this obtained basically a well-separated profile, except for some amino acid pairs, like, for example, the proline and phenylalanine, that are well reported in other CE applications to have identical migration times. Anyway, the use of MS data enables to clearly distinguish between these pairs. Then the standard amino acids were mixed with their isotopically labeled respective, and this mix was used to perform calibration cures for each component, as well as the determination of LODs and LOQs for all the amino acids, and they were all below 1 micromolar. First, we tried to monitor media from several vendors, and as you can see from the 3 electropherogram, it is clearly possible to distinguish amino acids content in each of them. Then we decided also to apply this tool for a real cell culture monitoring. So a CHO cell line was cultured in batch mode over 10 days, and aliquots were taken daily and analyzed. The graph here is just an example of the trend of leucine consumption over the 10 days of cell growth. And the overlay on the right side are the extracted ion chromatograms over the 10 days for leucine and isoleucine MS signals. So in the same way, this was done for all the amino acids, and it was possible to monitor their behavior along the 10 days with a very quick analysis in only 3 minutes. So in conclusion, on the ZipChip Q-Exactive platform, we perform 3 different workflows, and they all provide very accurate, in-depth, and quantitative data with a short analysis time. I hopefully gave an idea of the workflow that are possible to perform on this platform, and how this platform could also be flexible and is able to provide fast and robust analysis for some key workflows in biopharmaceutical characterization. In particular, I would like to mention the Q-Exactive performances in terms of resolution and mass accuracy that really can enable confident identification of native proteoforms. And with respect to samples direct infusion, the online separation of charge variant is surely simplifying data complexity, while the nanospray flow generated at the corner of the chip can really boost MS sensitivity. Microchip CE analysis surely has the advantage of low sample volumes that required, and this could also be important during drug development when the sample availability is limited. As well, the platform allows quick switching between applications, so it's really flexible and requires minimal method optimization. Just to conclude, I would like to thank you for the attention. Thank you, Thermo Scientific, for giving the opportunity to present this work. All my collaborators in the Characterization and Comparability Laboratory here in NIBRT, in particular our PI, Dr. Jonathan Bones, and the guys at 908 Device for the technical support and scientific advice, and all of you for attending this webinar. And I think now me and Ashley will be happy to take some questions.
Thank you, Sarah, and thank you, Ashley, also for setting this up so nicely. Yeah, we have a couple of questions if you guys are willing to do that. We have a few minutes left. There's one for you, Ashley. Sounds like it might be a customer for you. Somebody wants to know if you sell a ZipChip that fits on a Q-Exactive, not the Plus model, just the Q-Exactive.
Yeah, the ZipChip will fit on any of the QE mass spectrometers, as well as the Fusion Lumos series as well. So pretty much any Thermo.
Okay, what are the sample volume requirements for the system?
So I can take this one. The sample volume required for the system is there is a minimum of 10 microliters just to feed the autosampler then into the seed device in the microchip well. But typically, majority of these can be recovered because only between 1 and 5 nanoliter are consumed during the analysis itself.
Okay, thank you. And how difficult is it to optimize settings for monoclonal antibody analysis?
Okay, so as I showed during the slides, we mainly used only the fault methods that were inside the software. So there was really a minimal optimization that it could be like the field strength that I showed on the peptide mapping or the volume that you would like to inject.
Okay, we have one more question here. Can you verify that the field strength could be adjusted, and would you recommend lowering the field strength to improve the resolution? So it's 2 questions in one there.
Okay, I can take this. The field strength can be adjusted either as what you would consider a steady or field strength at a constant level, or as Sarah showed in the peptide mapping, the field strength can be applied to a linear gradient where you might want to lower the field strength at the beginning of an analysis, as in the example she showed, and increase it. And what that accomplishes is it maintains the peak width throughout the length of the run. So yes, it can be adjusted. However, by lowering the field strength overall, essentially you would be slowing down the entire analysis. So from a resolution standpoint, the way to improve the resolution would be to move from a shorter channel length, like the HS chip, to a longer channel length, whereas changing the field strength will merely affect the speed and not necessarily the resolution itself.
Okay, thank you. And you mentioned 5 mg per mil as the minimum concentration necessary for intact MAB analysis. Is it possible to go lower than that, do you feel?
Yeah, so I think it's possible. It depends on the numbers of features that you are really interested in monitoring. We found that with this concentration, we could really spot very low abundant one and some that we weren't able to see before. But yeah, absolutely, it's possible to go lower than this.
Okay, thank you. A couple more for Ashley. Will the ZipChip fit on the new Orbitrap Exploris 480, and will it fit on the SciX triple quad QTOF?
Yeah, the answer to both of those questions is yes.
Okay, excellent. Thank you for those. I think that takes us pretty much up to the half hour. So I would thank both of you again for that presentation and for your time today. We'll now close the webinar session. If there are any more questions that we haven't got round to answering, we will reply to you within the next few days. And as I said earlier, if you're listening to this webinar on demand, you can still use the question and answer chat box, and we will reply to all questions, although it may take a little longer to get around to it again. So thank you to our presenters, and thank you to you for attending. And remember, there are several more webinars in this series, so please come back and join us for the next one. Thank you all. Have a good day.